Exam 3, we will be talking lots of the things all together. First of all, we want to talk about is genetics and biochemistry related to the microbiology. And then we will have a guest speaker with Dr. Kenny talk about the meat safety and the HACCP. He will give you a tour, a very brief tour at the meat lab. And we are going to talk about the poultry meat processing, food safety afterwards. And then we'll have exam 3. So it's going to be the combination of different material. Okay, so the first couple of classes, today and Thursday, we're going to talk about genetics. So why are we going to talk about genetics? Because in the history, genetics is very important. It is an interdiscipline by the time between the biochemistry, biology, veterinary science, and genetics. It's all combined. When we talk about the microbial genetics, it's all started from DNA, RNA, all those things. And at the very beginning, it is coming from is an outbreak of a Spanish flu. So we will cover all this information. Now, the outline of this section we want to talk about is you already learned a lot called central dogma. So what is central dogma means? Which means the DNA, they're going through replication, and their genetic information will transfer to RNA, and this is called transcription. And transcription following by finally is RNA sequencing will become a protein. Now, of course, before that, they will have some biosynthesized amino acids first, then become a peptides. This process we call translation. Now we know that in certain bacteria, especially some of the virus, this thing could happen. RNA go back to DNA, that called reverse transcription. In the long term, it comes out with a PCR method. It's called reverse transcription PCR. Okay, so that's the story. Now we want to make sure it's easy, so we go talk about the replication, transcription, translation, the order like that. Because this is a general education class, so we talk about all these chemicals will be briefly. Now more important is we got to talk about history, why it becomes like that. There are lots of story behind it. Then we can learn from those early scientists what their research they are doing, from what type of the perspective they are doing the research to find out the DNA and the RNA and the protein sequencing, all those things. These slides you know, and I believe you already learned in your biology class a long time ago. So this is 1928. F. Griffiths did a very important and interesting mice study. So what is the story by the time? The story by the time is they going through a dramatic, tragic is 1912 Spanish flu. By the time, it's the same severe situation as COVID-19. It's more severe. It kills millions of people in the European country. So they try to developing is a vaccine by the time. When they developing the vaccine, they get to know a pathogen, which is we learned the Streptococcus pneumoniae. Between that, the scientific area also comes out with something. They want to find the genetic information. They call principles of transformation. They want to find what other genetic material it is, what's the genetic chemistry it is. Okay, so they started to do research with Streptococcus pneumoniae. Now, Streptococcus pneumoniae, we already learned that. It has a capsule. It's a diprococcus cell with a heavy capsule. They have two different types. One is called smooth. Another one is called rough. So it's very interesting. The smooth one is actually virulent, which means the mice will die if it's infected mice. The rough one, it's a surrogate, which means the mice still live. So the study, what they do, I use this as a mice, okay? First of all, they injected the mice with a smooth one. The mice is dead. Second group, they inoculated a mice with a rough one. The mice is alive. Now what they do, they inoculated a mice with a heated smooth one. The mice is survived. Now the next research group is interesting. The mice, they inoculated heated smooth Streptococcus pneumoniae, but with rough Streptococcus pneumoniae. What happened? The mice is dead. They got dead mice. What this tells you? This tells you the heated smooth Streptococcus pneumoniae and the rough Streptococcus pneumoniae, they absolutely have something transferred here. This fits the principles of transformation. But what it is, they don't know. It could be DNA, it could be RNA, it could be protein. They don't know about that. F. Griffiths worked really hard. He did all the study is on animals. So thousands of mice has been killed by the time. He's trying to do lots of the animal studies, but he didn't get successful because he get killed by the German bombing in 1941 during the Second World War. Okay, so 1941, the German bombing killed him. So, he didn't finish his work. But the time people knows there are some of the genetic information they get to do. So, 1928, the work is done. He did a lot of things following that, like 10 years, lots of the mice has been died during the animal studies. Then goes to 1943 to 1945. That is close to the end of the Second World War, but it's still everybody is busy. We get three great American scientists. Avery, McCarty, and McLean. They did this great study. I'm not going to go detail of this one. That could take another 10 minutes. Basic idea is that they know the genetic information could be DNA, could be RNA, could be protein, but really what it is. So they introduce an enzyme, which is could be DNase, RNase, or protease to destroy other chemicals, then verify the genetic information is DNA. And at the end, they get purified DNA chemical. That's what their job. So you can see what they do. They're using DNA extract, DNA transformation. Use DNase, no transformation. Use RNase, transformation happens. Use protease, transformation still happens. Which means the transfer information is not RNA, is not a protein. And later on they get purified. The chemical is DNA. So they know the genetic information is DNA. The transformation principle is DNA. This study is also a big progress compared to F. Griffith's study. F. Griffith's study we call in vivo, which means animal study, rely on and made lots of the mice. This is in vitro, which means they do not rely on animals. They do in the test tubes. It's a big progress in the scientific area. So now the thing happened is, they find that DNA chemical is a transformation important chemical. But other people has to verify that. This comes out 1952. A very important study called the Hershey-Chase experiment. This is Alfred Hershey and Martha Chase. This is not a candy bar, and that's not a Chase Bank. So you should know that. Alfred Hershey and Martha Chase. By the time Alfred Hershey is a professor at University of Oregon, Martha Chase is do something with her, like with him, like a research scientist. So what they do, they're from a different perspective. They using a bacteriophage. So why do they use a bacteriophage? First of all, bacteriophage, they can do two things. Either lysis bacteria, which means you are curing bacteria, or they will do temperate, which means their genome sequencing will be integrated into a bacterial genome sequencing. And very interesting to use bacteriophage because phage is a virus. And for the virus, the structure is relatively very simple. The inside, this is nucleus. So either DNA or RNA, never both. The outside is protein. Now, if you know the chemical, protein is always sulfite. For DNA is always phosphate. And sulfite, they have an isotope, which is S35. Phosphate also has an isotope, which is 32. Okay, so they can label those 35, this is 32. And this phage, go ahead to find is a bacteria. And when they find that, okay, once they go to the bacteria, the genome will be goes here. And then they will cause the lytic of the bacteria. And the phage itself, they use the bacterial information or material to biosynthesize their own phage. So it's very interesting when they do this, they combine. They use a kitchen blender to do just a blender, like you're making a smoothie. After making a smoothie, what they do, they did a kind of like centrifuge kind of thing, but what they find is that this portion is bacteria. This top usually is virus, because virus, the weight is relatively light. They find here, most of these are P32. None of them S35 is observed. What this means? Which means the bacteria phage carry on the genetic information going to the bacteria is P32, is not S35. And P32 is labeled as a DNA because phosphate is a very specific chemical for DNA. So this research from this standpoint, from the phage and the bacteria combination standpoint, verify that the genetic information is DNA. Okay, so you can see the slides looks like that. So then, we want to talk about something is, what is the DNA structure? What it looks like. It's very interesting, we can talk about that. And you already know. So we get to know the genetic information is DNA from two important studies. Actually, it's three. So then we get to know what is DNA. It is composed by like this. Okay? A complement with T, G complement with C. So running opposite direction, 5 prime to 3 prime, 3 prime to 5 prime. So what is DNA looks like? The DNA structure looks like it's very simple. When you draw, this is a 5-carbon sugar. We call it a ribose. Originally looks like this. What means D? Deoxynucleic acids. So you have 1 prime, 2 prime, 3 prime, 4 prime. And you go here 5 prime. This is gone. That's what we call DNA. The oxygen at 2 prime is gone. So that's a deoxyribose. It's a 5-carbon sugar. What you have here, this is a base. What are the bases we have? You already know. It could be purine or pyrimidine. What the purine looks like? It's usually a 5-carbon sugar, 6 carbon with 5 carbon. A pyrimidine, most of the time, is a 6 carbon. So what is purine? Adenine and guanine. What is the pyrimidine? Is cytosine and thymine. That comes with ATGC. And we also know A complement with T, G complement with C, and this middle we call it the hydrogen bonds. So we have a base, we have a deoxyribose. What else we have? That's a phosphate. So DNA, from some standpoint, they are composed by three major material: is the base, is the 6-carbon sugar, and the phosphate. So look at the phosphate there. That's very unique for the DNA. So from some standpoint, we can say the DNA is a very boring chemical because it only has base, deoxyribose, and the phosphate. Okay, so that's what the DNA. Now, what is this guy comes from? This we need to go to see 9053. This is everybody know about that. Is Francisco Crick and James Watson. Francisco Crick is a British scientist, is a physician, working in the Royal Community. James Watson is a student by the time at Indiana University. He has a hobby, is do the birds watch, like to watch the birds. After he got a PhD from Indiana University of Biochemistry, he go to study abroad at Cambridge University in the Cavendish lab, find Francisco Crick. They have about 12 years age difference, but they get along very well. They chat each other, they talk to each other every single day. They want to find the DNA double helix structure. So what is the structure of DNA? They try to solve the problem. It's very lucky by the time. In 1950, around the time, they find a very nice, great British scientist, a woman named Rosalind Franklin. Rosalind Franklin showed them her research results, which is in the textbook everybody knows. Use X-ray diffraction color photographer, whatever you call it. They find that the double helix. Rosalind Franklin, unfortunately, has ovarian cancer. So, their friends, Francisco Crick and James Watson, took her to have a trip in the European countries. In 1958, Rosalind Franklin passed away. That's the reason the Nobel Prize in 1962 did not award to her. Award to her boss, Maurice Wilkins. So in 1962, the Nobel Prize of the DNA, double helix, they gave to Francisco Crick, James Watson, and Maurice Wilkins. Now, another thing I want to point out here is that lots of the history, the biochemistry history, they're missing one important thing. It's very true. Rosalind Franklin showed them the X-ray diffraction double helix structure. But there is one thing. To let Francisco Crick and James Watson finally determine it's a double helix structure, is they get to have a lunch with a great American chemist, Sugaraf. So, Sugaraf shared them his research results. What is that? Everybody knows that. the ratio of A versus T and G versus C is 1:1. So what this means, in a different biological body, you may have a different amount of the A, different amount of the T, different amount of G and C, but the ratio is always 1:1. This is in the biochemistry area, is very famous Shograf rules. Once Shograf had a lunch with them, showed his results to them, they finally determined the DNA double helix C structure running opposite direction. They published this one piece of paper in the Nature. Only one page, but it is very important. The paper right here published in the Nature, they call this is the single most beautiful paper in the 20th century. Molecular structures of nucleic acids. It is only one page, you can see. And they find these results. They go to a pub. This is what the scientists they celebration when they had some discovery in the older time. They go to the pub. The pub is at Cambridge, suburb of London. If you go to study abroad or have a time to visit there, you will see that's called the Eagle Pub. In the Eagle Pub, they still have a plaque there, which is memorization. Francisco Crick and James Watson for their breakthrough finding of the DNA double helix. So they go there, they talk to the people, they pay them £2 to buy a beer and tell them what they find for the DNA double helix, but nobody really cares because nobody understands, but they are celebrated, share the big news with them. And that is the foundation of the modern biochemistry area. So the slides here you can see there is a base pairing which is arginine with thymine is 2 hydrogen bonds, guanine and cytosine with 3 hydrogen bonds in the double helix structure. So you can see very clearly here they are running opposite direction. Okay, one is 5' to 3', another one is 3' to 5'. And the connected bonds between every single unit is we call phosphodiester bond. So sometimes we always say the DNA strand running opposite direction, which means their sugar direction is different. Okay, so that's a DNA structure right here. You can see the 5' to 3', 3' to 5'. You can also see the picture for the replication. So we talk briefly about what's a DNA, what is the DNA structure. So next one what we want to talk about is the DNA replication. So let's move this out, let's talk about DNA replication. So DNA replication, look at the slides here. And we already know the conclusion is called semi-conservative. What I mean semi-conservative? Which means you have a double bonds. You always use one bond or one strand as a template to biosynthesize a new template and a new strand. So in the history, we had another very important study, which is called Matt Mendelson and Frank Starr study. So they're called Medelson and Starr research. So how they did that? This is the DNA double helix. They use these label N14. Nitrogen have the molecular weight is 14. Then they let the bacteria grow in the media containing N15, which is also an isotope. And you know that the DNA replication is semi-conservative. They use one strand as a template to biosynthesize another one. So what this means, once these two break out, you're going to find another one like this. So you have another one like that. So more and more will come in. So at the end of the day, what they do, they put in a centrifuge tube. And they find that at the bottom, with Sicilian chloride, when they do the centrifuge, they find that at the bottom, most of them are N15. At the top, N14. At the end of the day, more than 90% will be N15. N14 will be very limited. This research, using a very creative way to verify that, the DNA replication is semi-conservative. Okay? So that's the story behind it. Now we want to talk about what's a DNA replication, really talk about what this means. So we have to remove this. We want a little detail tells you how they created with each other. First of all, we talk about the bacteria. The bacteria DNA is always circular. Fungi and human being are a little bit different. Lots of the time it is linear. Circular, they always have the place to have the DNA synthesis. We call it a region of the replication. We also call it called the replication fork. And they started to have a semi-conservative replication. And at the end of the day, when they stop, they have the signal called TER, which means a termination. So how did it happen? We have two DNA strands. They are running opposite direction. The top one is 5' to 3' to 5'. The bottom one is 5' to 3'. They are running opposite direction. So first of all, everything happened is at this replication fork. They need something we call it initiation, which is DNA protein A. That is the initiator. DNA B protein, that is a holder. Then DNA C protein, which is we call it a helicase. So what this means? Originally, this is a double helix. We want them to be separated. So we need a helicase, which is an enzyme, to separate them. Now, because DNA is always twisted, so from the physics standpoint, it's very easy to come back and become double helix. So we need something to let them separate. Therefore, we need lots of the chemicals to attach to the side. This is called single strand binding protein. That's called SSB, to make sure they are separate. Now, how they started to do the DNA replication? It depends which bands. The top one is from 3' to 5'. The bottom one is 5' to 3'. So let's talk about top one first. DNA replication, you always remember, the direction is always 5' to 3'. Regardless of which bands, at the beginning, they all need a primer. The primer is RNA primer, so we need a primase to synthesize a small sequence. For 3' to 5', they could do naturally to 3'. So let's say we have A, T, G, C, G, C, T, A. So complementary we have T, A, C, G, C, G, A, T. They can do it very naturally. And they need the enzyme, DNA polymerase. They need DNA polymerase 1, okay, to do the polymerization. The function is 5' to 3'. But this bands, they get a problem. The top one, we call it a leading band. And the bottom one, we call it a lagging band. Why we say it's a lagging band? Because it goes from 5' to 3'. But the replication is always 5' to 3', so they get a trouble. So then what they have to do? They need to generate lots of those small segments. This is called Okazaki segment. These Okazaki segment is always synthesized by a RNA, small RNA. Once the Okazaki segment, they need to be connected. They need DNA ligase to connect it. Now, why the lagging band cannot do it naturally, like the leading bands to do? There, one of the theory is very easy to understand. Is if you look around these slides, you will see very clearly. From 5' to 3', they need a free available hydroxy bonds. However, if you're running opposite direction, this hydroxy end will not be showed up. So, simply to say, the lagging band, why they need to have an Okazaki segment? Because there is no 3' hydroxy bond. Or we say hydroxy residue, which means it's a free available one. But lots of the time they don't have it for the lagging band. So that's why they need Okazaki segment for the DNA ligase to put them together. So once everything is being set up, it's not done yet. So what happened is, let's say I have all these replication finished. We have some problem we need to solve. There are two problems we need to solve. Number one is topological issue. So what is topological issue? DNA is a double helix. There is, from physics standpoint, there is something talk about supercoiled. Originally, it is negative supercoiled. But when you just finish the replication, it is always positive supercoiled. Positive supercoiled is not a stable status. It's very easily to become back. So we need to let the positive supercoiled become negative supercoiled. And very lucky, we had the enzyme called topoisomerase. Topoisomerase, based on the molecular size, they have a topoisomerase 1, 2, 3, have a different one. Topoisomerase, very specific in E. coli, we call DNA gyrase. This is the first thing. It's called a topological issue. The second, what happened is fidelity issue. So this happened in the bacteria a lot. Ideally, the mother DNA replicated, become a daughter DNA. The daughter DNA and the mother DNA should be exactly the same sequencing. However, unfortunately, it's not true all the time. The reason is there some mistake happened. That's called a fidelity issue. So if we just finished the DNA replication, how much error it will happen? It is pretty high. 1 among the 10,000 base pair. 10 to the power of minus 3. This rate is very high, and we don't want that. So to solve the fidelity issue, we first of all have a tau is DNA polymerase 3. The DNA polymerase 3 could do is a proofreading. If you find anything is wrong, the base pair. So for example, we have ATGC, should be TACG. For something happen, this becomes A. The RNA polymerase could cut it and put a correct one back and then seal it. So that's called a proofreading. This is like you are writing a manuscripts or some class report. You need to do a proofreading. You need to make sure the spelling is correct. So that's the first thing that they do. Using proofreading, the mistake could be reduced to 1 divided by 10 to the 5, power of 5. So around 10 to the power of minus 5 or 10 to the minus 6. However, that is not good enough for DNA. Therefore, they have another one. They could use methylene. So often the time when we have 2 bands, ATGCCG, TACG, GA, for example. What happened is that at this end, we could be adding CH3 to fix it. Then we find there is a mistake there, we're going to cut it. Okay? Let's say this one we cut it, we become a C. And then re-add a correct nucleic acid base there. With the Messerian methods, we could lower down the error to 10 to the power of 8 or 10 to the power of 9. And this should be good enough. So from here, you can see what is the key thing for the DNA replication. It's very important is what happens at the replication fork. So this slide is important for you to do the exam because all of these enzymes are listed here. What are the function of these is the next important question. So we explained here very well. What is the DNA? What is the DNA B? What's the primerase? What is the lagging band, leading band for? Then DNA polymerase 1 to remove RNA primers, filling the gaps with DNA. Then Okazaki segments joined by the DNA ligase. That's we already talked about. And always remember these type of models they're using E. coli as an example. This is another important table here. You can see all those proteins in the column. On the right side, they have all types of the function. What are they? They describe the very much detail. So for example, what is a DNA B? It's a helix. They break down hydrogen bonds holding two strands of the double helix together, promotes DNA polymerase activity involved in the primersome assembly. So what is the DNA gyrase? You can see that. It's relief supercoiled DNA reduced as DNA strand as separated by the helices. So reduce the tension. SSB proteins, what's the function? Then see what's the function there. DNA polymerase is also here, and the DNA polymerase 3 is important. And here it specifically says that the proofreading to cut out the mistakenly mismatched pair, base pair is relying on exonuclease. Like a scissor can cut it. And we also have a DNA polymerase 1, and we talk about DNA ligase, topoisomerase, all of these are listed very well here for the function. So this table is important. And this tells you the replication machinery, how they do that. And we already mentioned at the very beginning, why the lagging bands need Okazaki segments, because they don't have a free available 3' hydroxy bonds. And here you can see this is what the DNA polymerase looks like. It's a hollow enzyme, it's also. Helicase, single-strand binding protein, topoisomerase, what are they? They all wrote detail. And these slides, I really like it, rocket right here. The figure here, which is described very well, the Okazaki segments, and how they happen here. And this is really good slides, and this is a very good picture to explain everything very detailed. And this tells you about the ligase, the Okazaki segments, how they connected. And this is talk about the DNA polymerase 3, how they do the proofreading. And it's very detailed. And they mentioned that this activity is not 100% efficient. Of course, we need to have other way to do it. Now, how we do the termination? When you see the TER signal reach the termination site, then the DNA replication will be stopped. And the topoisomerase will be temporarily break down the DNA molecule so the strands can separate. So this is the DNA. Okay, next one. We want to talk about the RNA transcription. So we want to talk a little simple. And the next class, we'll talk about detail of the transcription and translation. So let's start talk about RNA transcription. So we finished the replication. We're going to talk about transcription. So RNA transcription. So first thing first, what is RNA? What is the difference between RNA and DNA? We want to talk about something we already mentioned. Lots of the material for them are the same. That's the same. What's the difference? Several places. ribonucleic acids. There is O there, not deoxy. That's the first thing. Second difference, the base. What's the base of DNA? A, T, G, C. What's the base of RNA? Most of them are similar. The only difference, instead of you have T, you have U. That's uracil. The others are the same. Now, what else is different? DNA most of the time is double bonds. RNA most of the time is single strand. Another most important thing, DNA replication. You have error correction methods. We call it a fidelity issue, can solve it. RNA transcription, there is no way to fix it. If you have a mismatched pair and the transcription mistranslated, then finally the amino acids will be mistranslated. That could cause molecular disease. Lots of the disease happen like that because RNA transcription doesn't have that function to correct the mistake. Okay? So that's something I want to say. Now, what are the RNA we talk about? If we talk about the RNA, there's basically three different types of the RNA. First RNA is we call rRNA. That's ribosomal RNA. It's a structure. Second RNA is tRNA, transfer RNA. Transfer RNA is carry-on anticodon. The last one is mRNA. mRNA is carry-on 3-code amino acid sequence. So basically, we have three different types of the RNA right here. This slide is pretty good. They gave you a lot of the conclusion I draw on the blackboard already is guanine, cytosine, uracil is adenine. Those are the base of the RNA. The sugar is ribose instead of deoxyribose. The phosphodiester bonds is the same. We have three different types of the RNA: messenger RNA, ribosomal RNA, and transfer RNA. And most of the RNA are single-stranded. Some are double-stranded, but not that much. Okay, transcription. What means a transcription? Before that, we have the slides here. Talk about what's the function of the mRNA, tRNA, and RNA. So messenger RNA, we already said, carry the message for protein synthesis. tRNA carrying the amino acids during the protein synthesis, and the ribosomal RNA is components of the ribosome. So what is transcription? So we'll give you a very basic example. Okay? If I have a DNA right here, I'm rounding opposite direction. 3' to 5'. Another one 5' to 3'. Let's add the gene base there. This is A, T, G, C, C, G, T, A, T, G, let's say. So you should know the complementary bands of another DNA, the sequencing is TACGGCATAC. There's no problem. But what's the sequencing of mRNA? So first of all, mRNA is a single strand. They are complementary to this one of the bands because they use this band as a template. So this 5 prime, 3 prime to 5 prime. Let's go that way. So what's the sequencing? Exactly the same as the top. The only difference is instead of you have a T, you have a U here. So it's the same thing. A, U, G, C, C, G, U, A, U, C. That's the mRNA sequence. Therefore, for DNA, the top one, we call it a coding strand. Because the coding sequencing of the top one is exactly the same as mRNA, in the exception is U replace T. Then the bottom one, we call it a template strand. Now, for the RNA transcription, same as DNA replication. It is also a process of polymerization. Therefore, we also need an enzyme called RNA polymerase. This is a hollow enzyme. The detail we'll talk about on Thursday. Okay? So on Thursday, we will be talking about is 2 items together, which is transcription and translation. Okay. Not in, I want to remind you guys, do not forget you already being signed a sheet for you working group for the presentation. It will be started somewhere in the week of the 13th April, so about a month. So please, working with your group, find the time in the library you can start it to do. Don't make it very rough. Treat this as an opportunity to collaborate with each other, a good opportunity to do the oral presentation training. It is very important besides your exam, because I really want you to show you what you learned, like a self-learning, okay? So just to remind you, the study guideline is right here. I want to see your efforts. I know some of you not the major, science major, but try your best to explain those scientific stuff there. So don't forget about that. We put a very detailed guidelines here. If you need a paper for help, you can send me an email. I can try my best to get it for you. These are the listed topics, but not limited to that. And I tell you one thing very interesting. Last year, I have a student that presented, injected Clostridium perfringens to make the muscle relaxing for making a beautiful face. The student presented very well. So some of the topic is like a bacteria medical treatment topic you also could do. It's a very lots of fun. We can talk about that. And I don't want everybody go ahead and talk about the pathogen control. Okay, you're using lactic acid, you're curing bacteria. I don't want everybody to talk about the infectious disease, since we already talked. All the other topics, this you should really looking for. Antibiotic resistance is interesting. You can see lots of paper. Lots of good paper about that. And this FSMA regulation is also important. Since we are in West Virginia, we are very unique, have a small farms. It's a good topic. So you guys should talk. Also, emerging pathogen. I don't know all the bacteria or the virus. Like I said, only 5% we know. I know maybe like 80% of those 5%. So very limited. If you can give a topic about the new emerging pathogen from the news media or from your material, which will be great. So just to remind you, don't forget about this. You guys have to start to working. Okay, don't wait for the last day. Say tomorrow I present and I put everything together. We want you to be prepared well, and it's a lot of points there. Same as the exam. So be prepared for that. Okay. So thank you.